Hello everyone, how are you doing? I hope everyone is doing great. In this video I am going to explain defense mechanisms or body's defense systems. But before starting the lesson, I want you to subscribe my YouTube channel and make sure you turn on the notification bell and make sure you share it with your friends. So I'm going to start with defense mechanisms. Defense mechanisms are of two types. Okay, I'll try to make or use Kurdish in this video as well. ما مناعيتين مناعة specific ومناعة neo-specific. First, we must know what's the difference. Non-specific defense, it's an defense or immune response that is developed against no particular pathogen or antigen. So it can be developed against any foreign body, not a particular one. So this is called non-specific defense. يعني هر تشتكي non-self bit, foreign bit, the immune response is produced against. But yeah, specific, no. In case of specific defense, the immune response that's produced will only attack or act on the invading pathogen, a certain pathogen, okay, or a certain foreign body. That's not common or not a general immune response. Well, non-specific defense in this section I'm not in تفاصيلها طبعا. The speaker هي Robert Koch, the German doctor. He was the first one to identify a pathogen anthrax in cattle or cows. So he was the first person who developed scientific methods called postulates, Koch's postulate, in order to identify a causative agent of the disease, or what we call them pathogens. So what he did basically, he tested cattle. He compared healthy and infected cattle with anthrax. He collected blood samples from both of them. He could see that there is a road-shaped bacteria that's causing the symptoms to develop. And how did he prove that? When he isolated the bacteria from the infected cows and he ground them in his lab, and he made like solutions of those bacteria that were infecting the cattle. Then he injected, just in case of this experiment, he injected the healthy animal, okay, and the result was the healthy animals, they developed the disease. And when he collected blood from the newly infected animals, it was the same pathogen as in the first infected animal. As you can see, both of them are the same. So these are called Koch's postulates. In the general examinations, these postulates have come, احتمالا مثلا what is the first postulate in identifying a pathogen, or what's the second, or what's the final step or postulate that was developed by Robert Koch. So you have to memorize them in sequence. يعني بتنتو تسلسل برقي. It's no big deal. In the first step, scientific method that can be followed is the pathogen must be present in the infected animal but not in the healthy one. And that's clear, right? The second one is the pathogen that's isolated from the sick animal that has the symptoms must be ground in laboratory. And the third one, the ground bacteria, you can make a solution preparation of that infected or invader, and then you can inject a healthy animal in the lab and see if it can develop the disease. Then you will collect blood samples from the newly infected animal, that's the fourth one, and you will isolate and grow them in the laboratory. And the pathogen that was isolated from the first animal must be the same as in the last animal. So these are called Koch's postulates. I think that's enough explanation for this. Then what do we have? We have what pathogens, examples for diseases caused by pathogens. Pathogens can be viruses, bacteria, fungi, parasites, and even some invertebrates like some insects. Okay. In the exam, I must always remind you what's coming in the general examination, so you must have expectations. See, امتحانات وزاري مثلا أفيا هاتي أضليت فود اختيارات داين بسن أضليت فود كاتش نوعي باثوجيني أضليت فود شتكت. Is it a bacterium? Is it a virus? Or is it a protist or fungus? So you should know it's a fungus that causes athlete food because it's more common among athletes, and usually it develops in their feet, between the toes where there is high amount of moisture, especially when there is low personal hygiene. And the same for other diseases. I believe the pathogen names are given clearly, and the method of transmission. How are they transmitted? Because different pathogens are transmitted in different methods depending on the site of infection and the route of entry. Okay, this is no big deal. This is a part that some students ask us to do a video about the details, specific, non-specific defenses. How are you going to connect them and make the difference between. Okay, first we should know that there are three lines of defenses. We have the specific, non-specific. We have the specific, non-specific defenses, but they are grouped into three lines. We have the first line, second line, and the third line. I wasn't focusing enough. Okay, I was looking for this platform. Well, we have the first line of defense. First line. We have the second line, and we have the third line. You must know that the first and the second line, they can provide us with a non-specific immunity or defense, while the third line of defense, it makes up or provides specific immunity. Okay, let's see what are the components of these three lines of defense and the role of each component in making up the defense. The first line of defense consists of what? We have skin, okay, and mucous membranes along with their secretions. Skin and mucous membrane, students, they make up the first line of defense. They make up a physical and a chemical barrier. And this question has come in the general examinations. Both of them, they make a physical barrier. Skin, our body surface is covered by skin. An intact skin, like a skin, a healthy bit, a normal bit, it's less likely for a pathogen to cross it. Same as mucous membrane, but internally. Mucous membrane, مثلا in nasal cavity, internal air pathway, digestive system, blood vessels. So these two membranes that are epithelial tissue, and epithelial tissue, the cells are tightly packed. No or few or gaps are left between the cells, so it makes quite a physical barrier. When we say a chemical barrier, it's because both skin and mucous membrane, they have secretions. For example, in case of skin, as you can see here, let me label it. There are sweat glands, oil glands, and waxes, for example in ear, So these secretions that are released by glands and skin cells, they provide a chemical defense. An example is the presence of an enzyme called lysozyme, which is found in sweat or in tears. It targets infectious bacteria and destroy them. It's lytic. It causes lysis to bacteria. Mucous membrane, it also provides a physical barrier, okay, but especially interior surfaces, okay. Interior surfaces, as you mentioned, like in the air pathway, in the blood vessels, and so on. They make a physical barrier and also a chemical barrier as they release mucus, right? They release mucus, and mucus is a sticky substance that can trap or capture pathogens, and usually the area where mucus is secreted, there are cilia bearing cells, and where is cilia جهي, the cilia, it's mentioned here, the cilia will sweep the pathogens trapped in mucus along with mucus to outside, for example to the throat, to be swallowed into the stomach, and then the pathogen will die in stomach acidity. Okay, so let's not forget, one of the two main components of non-specific defense are skin, mucous membrane, and their secretions. This is the two major components. Then another let's say part of the non-specific defense is inflammatory response. Guys, يعني تفيت انتو بظاني component non-specific defense, specific defense chana, all the details, because in general examinations, if you have noticed, there are many questions about the components of specific non-specific defenses. They will tell you, for example, which one of the following is not belong to a specific defense, or which one of the following does not belong to non-specific defense, or they will bring you a list and they will tell you, they will maybe give you exceptions. So in all cases you should know the components of both types of defenses. Okay, here we have inflammatory response. Inflammatory response, which means inflammation. We keep saying inflammation happened, and so inflammation is basically a response that develops against all pathogens. Once a pathogen is able to get crossed by skin and mucous membrane, an inflammatory response can develop. And we said an intact skin or mucous membrane, it's less likely for a pathogen to cross it. So usually inflammation happen in case of injuries, internally, externally. So an injury, as you can see here, an injury in the skin or in the mucous membrane can allow pathogens on the surface or environment to enter the tissue. Okay, so this is how an inflammatory response develops, or inflammation. So the first thing that's needed for an inflammation to develop is the presence of pathogens. And by the way, this is one of the questions that have come in general examinations. It says which cells are needed for an inflammatory response to begin. You cannot say phagocyte or damage cells. No, they're pathogens. So the entry of pathogens will cause the damaged tissue. You see here, the damaged tissue or cells to release a substance, a chemical substance, chemical messenger. It's called histamine. Histamine. It's mentioned here. Now I am labeling, if you see where I'm labeling. So histamine, it's basically a chemical substance that causes attraction of phagocytes to the site of infection. How? By increasing blood flow to the site of infection, and also it increases permeability of capillary wall, as you can see here, capillary wall becomes more permeable to phagocytes to leak through. We already know that white BCs, phagocytes, they can change their shape. They can squeeze through the capillary walls. You have studied this in circulatory system, blood section. Okay, so they will go to the site of infection. They are phagocytes. They have the ability to phagocytosize pathogens. And once pathogens are phagocytosized, they will most likely die inside the phagocytes. So they will be destroyed. What are the symptoms that can develop in the site of inflammation or when inflammatory response develops? This is important. The symptoms are pain, redness, swelling, right, and warmth. Here, these are the symptoms that develop in the site of infection. And again, what causes the attraction and increase the permeability of capillary wall? It's a chemical substance called histamine. How is histamine released, or what are the histamine producing cells? They are the injured or damaged cells. Okay, sorry. So we did mention that phagocytes will go to the site of infection, but phagocytes, we should know what are phagocytes. Phagocytes is a group of white blood cells that have the ability to engulf pathogens or maybe cancer cells. There are two types of phagocytes. We have neutrophil, which is the most abundant type of phagocyte. This is very important. It has come in the general examinations. And also macrophage. So these two, both of them are the two types of phagocytes. And both of them can attack pathogens. Okay? They attack pathogens, they engulf them, and they break them down in their inside. You know, there is a process called phagocytosis and the fusion of phagosome with lysosome, and there are hydrolytic enzymes in the lysosome that break the bacteria or the pathogen down. Okay, another cell that plays an important role in non-specific defense are natural killer cells. So natural killer cells are also white blood cells, but the difference is, or what differentiate natural killer cells from phagocytes is that natural killer cells, they do not target pathogens. Not pathogen. What they kill, what they attack are pathogen infected cells. Usually pathogen infected cells are viruses, like viral infected cells, because only viruses enter the cells, but other pathogens, no, they don't need, or they may not be able to enter the cell because viruses are too small and they cannot survive outside the cell. They need to use cell's machinery system for their replication, metabolism. Okay. So natural killer cells, they target cancer cells and pathogen infected cells. These are the two cells that are most often targeted by natural killer cells, and this has come in the general examination. What cells or which one of the following are targeted by natural killer cells? So two types of cells: cancer cells and pathogen infected cell, in other words virus infected cells. So you should make the difference between phagocytes, which are neutrophil macrophage. They target pathogens, okay, so they wait for pathogens, but natural killer cells, they do not attack pathogens themselves. Instead, they attack pathogen infected cells or cancer cells. I hope you understood this part. By the way, if you have questions, do not hesitate to write them down in the comment, okay, under this video, or send me in private. Okay, so we did mention the role of skin mucous membrane and the secretion they release, and then inflammatory response, and then the role of phagocytes. And by the way, inflammatory response including phagocytes, natural killer cell, and even what coming next, they make up the second line of defense. But skin and mucous membrane and their secretions, they make up the first line of defense. Anyways, all of these are non-specific defenses, whether first line, second line, all of these parts, mechanisms, and cells, they make up the non-specific defense part of immune system. So now we have the temperature response, or temperature change, or what we call it fever. Okay, you know our normal body temperature is 37. Okay, I may use some different colors. Okay, so our body temperature is stabilized at 37, okay, Celsius degrees, but an infection can trigger fever. When we say infection, it means a pathogen entry can trigger fever. Okay, there are two types of fever. A fever could be moderate, يعني اگر تايقا يا mild bit تايقا متوسط bit, like between 37 to 39, that's okay. It's good. It's actually good, because this fever, it plays two roles: slow down bacterial growth, okay, the invaders, and promote white blood cell activity. So a moderate fever is quite useful. It will slow down bacterial growth, and also it will activate or promote the activity of white blood cells to take a response. But a very high fever that's greater than 39 Celsius degrees, it can be dangerous. Why dangerous? Because it can destroy our body proteins or cellular proteins or enzymes. This has come in the general examination saying like, how can a high fever become dangerous? In the options, instead of cellular proteins, cellular enzymes are given. It's the same, because enzymes are proteins, and so they can denature at high temperatures above their optimum temperature. Okay? So fever or temperature response is another part of non-specific defense. Let's not forget. We have to, summarize all the parts and components of non-specific defense. What else? We have some proteins, okay? Some proteins that also provide non-specific defenses. A group of them that circulate in blood are called complement. So complement proteins. And by the way, complement proteins, they can also be found in specific defenses. We will take that in the next section. So complement proteins, they provide specific and non-specific defenses. So when they encounter a pathogen, they can become active, okay? But the important thing is that how do they kill the pathogen? They make a ring-shaped structure. This is very important. They make a ring-shaped structure on the cell membrane of what? Of infected cells, and causing them to die. So let's not forget, complement systems. The complement system, they target infected cells, okay? They puncture their cell membrane. They cause puncture, it means make openings, holes. They puncture their cell membrane, and the cell will die from the loss of fluids inside. It will shrink, and plasmolysis will happen. Good. Another protein that is released by infected cells, it's called interferon. Interferon, students, بروتينك أو سيلة فيروسي infected كرين, يعني virus infected cells, they release this protein as a signal in order to signal the nearby cells to take precautions, make defense mechanisms of their own. In other words, to develop resistance. إذن اف interferon proteinaka أو cellت virus هاد نافدا. Okay, if we have a cell that is infected with a virus, this is a virus infected cell, and there are healthy non-infected nearby cells. So the virus infected cell will release proteins called interferon, and these proteins will signal the normal cells in order to develop resistance, either by producing defensive proteins, enzymes, or whatever can give them the chance of survival. So that was all about the first section. If we summarize non-specific defenses, we have two lines from non-specific defenses, one line specific defense. Non-specific defenses include skin mucous membrane and their secretions. So you should know, for example, oil, wax, what else, sweating, and the lysozyme. All of these, they are the secretions and enzymes that are produced by skin and mucous membrane. So all of these are non-specific defense, make up the first line. Then we have when the pathogen is able to cross skin and mucous membrane. Of course, it will go into the tissue, and the damaged tissue can release chemotactic substances or chemical messengers called histamine. So histamine also is a part of non-specific defense. Histamine, we did mention how it causes increasing the blood permeability or flow to the site of infection, permeability of capillaries to white BCs, and we did mention the role of phagocytes, macrophage, neutrophil in phagocytosis, engulfing pathogens, and also natural killer cells can attack virus infected cells or cancer cells. And also the temperature changes. A moderate fever can be useful, slow down bacterial growth, promote white BC activities. And then we did mention the role of proteins as it's shown here. There are two major proteins or groups that provide non-specific defenses. One is complement protein, and the other are interferons released by virus infected cells. So all of what we have explained so far are components involved non-specific body defenses. Now we go to specific. Okay, specific immune defenses. See, it's mentioned here. The third line of defense, it's all specific. Okay, specific immune system. It is provided by a group of organs, tissues, and cells. Okay, what organs they play a role in specific defenses? I think that figure will tell us the answer. See, there's a group of organs, lymph nodes that play a role in specific defenses. Here we have adenoid and tonsil in the head region. There is thymus. By the way, thymus, it is an organ where T lymphocytes complete their development, and there are multiple lymph nodes found in many body parts, including axillary, pelvic area. If you remember the role of lymph nodes in lymphatic system, after the tissue fluid is collected by lymphatic vessels, it's filtered in lymph nodes, because in lymph nodes there are pathogens, and pathogens will attack. There are white BCs. The white BCs in lymph node, they will attack pathogens when they enter the lymph node. The largest lymph node, it is the spleen. Okay, spleen, it is the largest lymph node, and spleen, it's like a house for red blood cells where functional red blood cells are stored and aging red blood cells are broken down. It's the largest lymphatic organ actually. Even there there are lymphocytes that can attack pathogens collected from the lymph. Also we have bone marrow, another organ of the immune system. Okay, you already remember the role of bone marrow in producing blood cells, especially red bone marrow. So those blood cells, including white BCs, that play a role in defense. Okay, so these were the organs and tissues that are involved in specific defenses. But we did mention there are also cells that provide specific defenses. Only one group of cells can do so. We have lymphocytes. Okay, including B lymphocyte, T lymphocytes, they can provide us with specific defenses. But let's not forget, there is also macrophage that can get involved in specific defenses, and I will explain later. So now lymphocytes, they are the major cells that provide us with specific defenses, and these lymphocytes are produced in the bone marrow, but then they travel to other organs to complete their development. For T cells, they go to the thymus. That's why they are called T, from thymus, the first letter. T cells, they go to the thymus to complete their development after they are produced in the bone marrow. But B cells, most of them, they stay in the bone marrow. Okay, they complete their development in the bone marrow, and some can travel to the spleen to complete their development. Okay, and these are the organs we mentioned, the role in immune system. I have made them like a table so that you can connect them and summarize them all together. So since we are talking about these specific defenses, so there must be something that makes this type of defense to be specific. What determines the specificity? It is the presence of certain substances, proteins or sugars that are found on the surface of pathogens. Those are called antigen. What is an antigen? Antigen is any substance that our immune system recognizes it as non-self, or it's not a part of the body. So it could be proteins, sugars, anything found on the surface of pathogen, or they could be, for example, pollens. They could be some chemical substances. They could be toxins. Okay, so there are many things that can act as antigens. Anything that's recognized by our immune system as foreign, non-self, بجنبي antigen. Okay, so as you can see on this figure, these are pathogens, and different pathogens have different antigens. Even antigens found on the same pathogen can be different. Although in the figures shown, the antigens that cover the surface or are found on the surface of pathogens, all of them are of the same type. See them? But in some pathogens, there could be more than one type of antigen. But on the surface of lymphocytes, okay, whether T lymphocyte, B lymphocyte, there are specific proteins called receptor proteins. These receptor proteins, they are all of the same type. You cannot find receptor proteins located on the same type of lymphocyte to be different. Never ever. So all the receptor proteins found on the same lymphocyte, they are all of the same type. But antigens found on the same pathogen, they can be different. Okay, so that's why we call it specific defense, because the type of lymphocyte that is stimulated, okay, to develop an immune response, it depends on the type of antigen that has entered the body, okay? So depending on which type of antigen has entered the body, only the lymphocytes with complementary receptor proteins to that antigen will be stimulated. And they will answer this. You will know why are they stimulated in the next pages. So you must know that antigen and receptor proteins, they must be complementary in order to bind together, and only the binding can trigger other immune cells to take a response. So there must be a complementary shape, okay? So again, the specificity of immune system, it depends on the type of antigen that's found on the surface of pathogen, or even if they were free antigens. So that will determine what type of lymphocytes to be stimulated. Examples are given again for antigens. It's not necessary to be only molecules found on the surface of pathogen, but they could be toxins, pollens, insect venom, and so on. Even when a tissue or blood from incompatible group is transfused, this can work as an antigen. You remember the blood grouping system, that transfusion of an incompatible blood group, okay? The red blood cells of the donor, the antigens found on red blood cells will work as antigens in this case, because they will induce an immune response in the blood of the recipient if it's not compatible. So we did mention how antigens are identified, as lymphocytes have receptors, and these receptors are complementary, okay? Complementary, they can have a complementary 3D shape to that of antigen, and so they can bind to them and an immune response can begin. Let's see how does that work. So see, an example is given here, students. An immune response that is developed against one pathogen like cold virus cannot work against another pathogen like flavirus, because cold virus, flavirus, they have completely different surface antigens. So lymphocytes that are stimulated against cold virus, they can only attach to cold virus, right? Because they have only protein receptors that are complementary to cold virus antigens. But flavirus is quite different. Another immune response is needed. Okay. Now let's explain how does specific immunity works, the lymphocytes, okay? I will use the figures as much as possible because that's the only way that you can digest all the details easily. Otherwise it will be sort of complicated. Okay. So I'm the hamia figure green and dako يعني تأمجك habit for all the process to connect all of them together. So once a pathogen is able to cross the first line and the second line of defenses, in other words, once a pathogen is able to escape from non-specific defense mechanisms, they will encounter the specific line of defense, okay? The first cells that play a role in specific defenses are macrophage, okay? Although macrophages, they do not recognize a particular pathogen, okay? So they do not, they do not have specificity to attack what type of pathogen. No, they target all pathogens. but they are important for specific defenses as well, and you will see how do they work in specific defense. So for example, in this case we have a virus, and as you can see the virus is covered by many antigens. Okay, so when this virus is engulfed by this phagocyte, which is a macrophage, the macrophage will break it down in its body, okay, inside. The antigens, after the virus is broken down, the antigens of the virus are released or are presented on the surface. As you can see, you see those green little spikes. They are the antigens of the virus. After the virus is digested, now the antigens are presented on the surface of macrophage, and that's why macrophage is called antigen presenting cells, okay, because they can present antigens of the pathogen. That's not our story. So the presence of these antigens, why do they expose these antigens? It's because they will expose them to other immune cells to come and help. So there is a group of cells called helper T cells, which means they are T lymphocytes. Okay, they help in specific defense pathways. Helper T cells, just like any other type of T cell, they have receptor proteins. These receptor proteins, they are complementary to the antigen presented on the surface of the macrophage. Okay, you are wondering, how does that occur? As we mentioned earlier, the type of antigen that's found on the surface of pathogen or anywhere, it will determine the type of lymphocyte that's going to be stimulated. For example, the type of helper T cells that are stimulated in this case, they have complementary receptors to these antigens, and that's why only this type is stimulated, not other types. So once the helper T cell is bound to the antigens presented by macrophage, this will cause the macrophage to release a protein or cytokine called interleukin 1. Interleukin 1. What does interleukin 1 do? Interleukin 1 will target helper T cells, will stimulate them to release what? To release another cytokine called interleukin 2. So interleukin 1 is released by macrophage, okay, and interleukin 2 is released by helper T cell. So interleukin 1 will target helper T cells, activating them, causing them to release interleukin 2. Interleukin 2 has 3 target cells. Interleukin 2 can target other helper T cells and stimulating them to undergo further division, like to increase the number of helper T cells. This is the first target. The second target of interleukin 2, as you can see where the arrow is marking, is another group or type of T cells called cytotoxic T cells. From the name cytotoxic T cells, they are quite toxic to cells, okay, to certain types of cells. These cytotoxic T cells, they also have receptors, protein receptors, okay, and they are stimulated because of the release of interleukin 2. So cytotoxic T cells are produced in response to interleukin 2. What is the target of cytotoxic T cells? They target, just like natural killer cells, they target pathogen infected cells, cancer cells, and even sometimes parasites. Okay, these are the targets of cytotoxic T cells. In this case, we have a virus infected cell. As you can see, this is a virus infected cell. The greenish molecules are the antigens of the virus. After the virus is digested, the antigens are presented on the surface of the cell, just as in case of macrophage. Now this is an infected cell that will die anyway. So, you know, the virus can only survive within the cell. The virus, they don't want to kill the host cell they are living inside. But when the cytotoxic T cell comes, it will digest or kill the infected cell, so that the virus will also get killed along with the infected cells, because it's their host. It's what provides them with nutrition and enzymes needed for replication. Okay, I hope it's clear so far. By the way, the specific, okay, let me use a different color. What you see in this figure, this part, it is called initiation of an immune response. It's just initiation. How does a specific immune response begin? Okay, then step 4 and 5, they are called cell-mediated immune response. Cell-mediated immune response. Why is it called cell-mediated immune response? Because it's mediated by a group of cells called cytotoxic T cells. And let's not forget, so far, helper T cells, they have played a role in cell-mediated immune response, okay? Because they release interleukin-2, and then interleukin-2 stimulated cytotoxic T cells. Okay, the other type of, or the other pathway of specific defense is called humoral immune response, or sometimes called antibody mediated immune response, because you will see at the end antibodies are produced. Okay, so we did mention that interleukin-2 that is released by activated helper T cells in response to interleukin-1, it targets 3 types of cells: helper T cells, cytotoxic T cells, and moreover interleukin-2 also will target B cells. Okay, B cells. B cells is another type of lymphocyte, as we mentioned earlier. They also have complementary receptor proteins, and so only those B cells are activated that have complementary receptor proteins to the antigens found on the surface of invader virus. Okay, not other types of lymphocytes can be activated in any of the 2 pathways. Only those lymphocytes, whether B lymphocyte, T lymphocytes, are activated with complementary receptor proteins on their surface. B cells will undergo further division, like in response to interleukin-2. B cells will undergo further division sorry, and also they will undergo differentiation. So they will increase their number, and they start to differentiate into 2 types of cells. Some of the B cell, remember this term, some of them, not all of them, some of them will develop or differentiate into memory cells. And memory cell, they provide us with long life immunity. They can remain in the body throughout the life of individual. We will explain the role later in vaccination. So this is how memory cells are produced, and memory cells, you know, they are actually making the principle of vaccination, and they are the basis of the activity of vaccines, because memory cells, after secondary infection with the same pathogen, they can recognize the pathogen directly, and then they can activate other B cells to produce plasma cells and then antibodies in large amounts, and they will they will attack the invader, because they have even the chance to outnumber themselves. So we did mention a term, secondary immune response. By the way, primary immune response, all what you are explaining now is primary immune response. When you get infected, for the first time with the pathogen, and your immune response, the first immune response that develops against the pathogen is called primary. But when you get infected with the same pathogen for the second time, memory cells are present. They will start the secondary immune response by stimulating other B cells, producing plasma cells, and so producing large amounts of antibodies. So B cells can differentiate into memory cells, and they can also differentiate into, as you can see, plasma cells. These are plasma cells. So even plasma cells, there are many types, and what type of plasma cell will develop, again, it will depend on the type of antigen. Okay, so the plasma cell, the type of plasma cell that is stimulated or that's produced, it is in response to the type of the specific type of antigen, and that's why specific antibodies are produced. And all the antibodies produced by plasma cells are the same, because the plasma cells that are stimulated and produced, they are also the same. They are said to be clone. A clone of plasma cells is produced. Okay, so the antibodies, then they will attack the pathogen, but there's one important point. Antibodies, they do not kill the pathogen directly. Okay, antibodies, they do not kill the pathogen directly. There are mechanisms of antibody activity we will explain later. So this part, students, all this part from step 6 to the last step, we call it humoral immunity or antibody mediated immunity. So if we sum up all this up, okay, see the picture. So specific immunity, it starts with the macrophages. At first initiation, again, sorry, I believe I caught a cold. Memory cells are part of the the second pathway. There are two pathways. There is cell mediated and humoral. So the first one, at first we set initiation. At the very beginning, the macrophage will engulf the pathogen and then will present the surfaces on its surface, and then the exposure of antigens داد بجرب كما كردي دب كما كردي دا قالك دنا جشت بيت sorry. إذن أول step چيا macrophage the pathogen engulf كات the antigen te present كات سر surface خو ديو دا helper T cell دهن they have complementary protein receptors دشن خب في antigeni وگرن وقت binding چه بو اکثر the macrophage activate بيت the interleukin 1 release كات interleukin 1 ده چكات interleukin 1 target هاي بتني helper T cell the activation بو other helper T cell چكات باشا. So in order for helper T cells the coach release كان cytokine جديد بجنا interleukin 2. Interleukin 2 say target it can target other helper T cells to undergo further division, and also it can target cytotoxic T cells and B cells. So it can target cytotoxic T cells, so that cytotoxic T cells to be stimulated and produced in large numbers, and so they attack cytotoxic T cell دچن هرشي كان سر pathogen infected cell or cancer cells. و mechanism يواضحت VNA دا child cell adhesion. They make a hole in the cell membrane. This causes cell burst because of fluid loss. Target دیا interleukin 2 ما قلتي B cell B cell activate كات B cell the divide بن و the differentiation بو چبيت عندك شوانا دبنا memory cell ما قلتي memory cell ده منا جسمي ده they will provide us with a long term immunity. اگر هاتو جارا دوه نفسي pathogen هاتت جسمي ده the memory cells will respond quickly, and they will stimulate other B cells and plasma cells to produce large amounts of antibodies. ما قلت B cell اتبيتن هندك شوى اتبنا memory cell, some of them, but most of them دبنا plasma cell or the antibody or chicken. وما قلت the antibody will destroy the pathogen indirectly, not directly. So I hope all these mechanisms were clear. I have made summaries for you because these are very important, very very important. يعني الجنرال examination بسيارة وزاري تشوتت مثلا دي بشتتا which one of the following cells, okay, is not a part of cell mediated immune response. So they will give you these three and another option that you can choose. On which one of the following cells they are not a part of humoral, or they are a part of humoral, or they are a part of specific, non-specific. On مثلا دبوتاينيتن إكجوان سيلا دبستتا which one of the following cells belongs to non-specific defense. which دبوتاينيتن non-specific defense دبوتا مثلا سيلاينيت, you have to know all these cells, they belong to specific. On مثلا primary secondary immune response هذا همي تبعي specific باشا primary secondary هذا همي specific. الفيراجي مسامر يكوديو هاي specific non-specific defense their components, so that's very important. ما بحث primary و secondary كر ما قلت primary اما وقت دا سبيك pathogen تد او مناعة تشد بيت او همي primary. طبعا أنا بحث specific كاين هذا همي specific. secondary اگر هاتو بجرد ويتو infected by with the same pathogen ما قلت memory cell ده شول كان و يعني أو stipid بري تشبونا memory cell, they are not needed. So memory cells can produce they can act quickly, and they can stimulate other B cells to produce large amounts of antibodies in a short period of time. Well, vaccination ما قلت it depends on memory cells as بجمع في موضوع بصناعية, so I don't have to give details. هدفنا وباز بفيديو specific و non-specific شرح كان. I believe the other parts are simple. If you have questions, you can, as I told you, you can ask me in comments. Again, I kindly ask you to subscribe the channel, share the video with your friends so they can get some benefit at least. I hope you'll find it useful. Inshallah, sarkaftiban. And whenever you have questions, I'm always available. Thank you so much. Have a good time and all the best. Bye bye.